Hey guys, today we're going to talk about protein synthesis, and this is basically how your body makes proteins. So it says, what do these Chinese symbols say? And it gives you the symbols, and then it says transcribe to English alphabet, so they transcribe it, and then it says translate it to your English words. So that's pretty much the way that making proteins takes place. You have your DNA strand, your DNA information that is in your nucleus, and on that DNA there are genes, and those genes are basically telling the cells how to build specific proteins. So basically everything in our body has a certain genetic code. And so DNA carries that genetic code. So it needs a messenger because DNA cannot move from the nucleus to the ribosomes where protein production is located. So we basically have something called messenger RNA that is made in the made in the nucleus, and then it goes out through nuclear pores to the ribosomes in order to make your protein. So RNA is a nucleic acid just like DNA, except that it has a couple of differences. One of those differences is that instead of deoxyribose sugar, it has ribose sugar. And another difference is that RNA is single stranded, not double stranded. And the last difference is that RNA contains the nitrogen base uracil, which is represented with a U, instead of thymine, which is represented with a T in DNA. So instead of adenine bonding to thymine, it bonds to uracil. So here are some of the common differences between the two. You have your DNA, which is double stranded. It contains thymine, it contains the deoxyribose sugar, and it's found only in the nucleus. Your RNA is single stranded. It contains uracil, it contains ribose sugar, and it's found in both nucleus and cytoplasm. The things that they have in common is that they're both made up of nucleotides, which means they both have a sugar and a phosphate group making up the sides or making up the base connection of their structure. And then they also have nitrogen bases that are attached to it, to the sugar. And so they both contain the bases adenine, guanine, and cytosine. So does this diagram represent DNA or RNA, and how can you tell? So one of the ways that we can tell is that we have two sides of sugars and phosphates. So that would mean that it would be double stranded. So therefore, it is DNA. Another way that we can tell is that one strand is going from bottom to top and the other strand is going from top to bottom. And remember that with DNA, we have those opposite strands or anti parallel strands that are taking place in the creation of a DNA molecule. So there are three types of RNA. There's messenger RNA, which is called mRNA. There's ribosomal RNA, which is rRNA. And there's transfer RNA, which is tRNA. There's also actually a fourth kind, which is called RNAi, but we're not going to talk about that one right now. So messenger RNA starts out in the nucleus. It makes a copy of itself from DNA. And so there's the DNA bases. The RNA bases line up with the DNA bases in order to make their strand. Then the messenger RNA, when it's completed, will go out the nuclear pores of the nucleus into the cytoplasm and then travel to the ribosomal RNA. So ribosomal RNA is little r RNA, and this makes a large part of the ribosome. And so it reads and it decodes the mRNA as it's being processed through it. The last RNA is called transfer RNA or tRNA, and this is what brings in the amino acids to the ribosome. They're joined together, those amino acids do, and they form the protein. So protein synthesis is basically taking those amino acids that the tRNA brings in and it joins them together in order to form that protein. There are two steps that occur in order for protein synthesis to produce the protein. So the first one is called transcription. And transcription is just basically where you're copying the DNA and you're making an mRNA. You're transcribing the DNA. So therefore, you're making that copy of mRNA or messenger RNA. Translation is where you actually take the messenger RNA or mRNA and you read it. You find out what amino acids it's coding for and how those amino acids are going to connect in order to form your polypeptide chain, which is your protein. So transcription occurs in the nucleus. It creates the mRNA, which is a single strand of what DNA is reading. So it's a complementary copy of DNA. So how transcription works. So if you look at the bottom, you've got your DNA and you've got your mRNA. So DNA is going to split. If you look at the picture at the top, right hand corner, and it exposes a site, and that site is called the active strand. And then you have a complementary mRNA, the nucleotides line up opposite the active strand, and that forms your mRNA. So in the picture, the blue is your DNA and the pink is your mRNA being made. So then mRNA leaves the nucleus and goes into the cytoplasm, then goes into the ribosome, where translation occurs. So translation occurs in the ribosome, and all three types of RNA work together during translation to produce your proteins. So you have on your mRNA strand is sort of like the instructions of what amino acids will be linked together to produce that polypeptide. So your ribosomes decode the instructions by using what we call codons. So the codons are located on your messenger RNA. 3 bases will code for 1 amino acid, and the different ways that the bases are arranged code for different amino acids. So each codon is matched to an anticodon, which is on your tRNA, and that determines the order that the amino acids will link up. So here are your codons in black, and here are your anticodons in blue. And then your amino acids that are attached at the top. So you can have 2 different types of codon charts. The one that you most commonly see is the box. But there is the one that looks sort of like a sun, and actually it's a little bit easier to use. But the codon chart is used to determine the sequence of amino acids in the polypeptides. The set of 3 messenger RNA bases or codons are used to find these amino acids. And the way that you do that is if you had AAA as your code, the first base would be A on the side. Then you would go to the A on the top, and you'd have where they come together in this box, in the third box from the left. And then you would find the A on the right-hand side and go over, and lysine would be what you would code for that particular code. If you're using this box, you would start with your A, you go to your second letter, which was A, and then your third letter, which is A, and then it would be also lysine would be what popped up. So here's an example of how you make your mRNA and you get your amino acids. So remember that U replaces T in mRNA. So wherever you have an A, you're going to have a U for your mRNA. So for your DNA here, you have a TAC. That translates to mRNA as AUG. And then you have GCA in DNA. That translates to CGU in mRNA. And then you have TGG, which translates to ACC, and then AAT, which translates to UUA. So if you look at the first letter for that mRNA code, you find A. And then you're going to go over to where the A and the U meet, which is beside it, the box right beside it. And then you go to the right-hand side, find your G, and you come back over, and it's methionine or thionone. And so you basically will always start out with that code. You usually have AUG as your start code for most of the proteins that are formed. Then you can translate the rest of them. So the next one is C. You go over to G, and you find your U, and it's arginine. Okay, and then they've translated the other 2 for you to make your protein. Usually you'll come to a stop sign at some point. You'll see a few stop codes, and that's where your protein would be finalized and be completed. Okay, so I hope that helps you understand a little bit of the information about protein. um, synthesis and how we make our proteins. At the end of the week, you're going to be making an alien. That is basically how you are going to code for them, is by making the mRNA and then finding the attachments of the amino acids.